Welcome to this tutorial for Simon's design in nQuery. In this tutorial, we will cover how to calculate the sample size for Simon's design by looking at an example from the Journal of Clinical Oncology, which evaluated a targeted therapy for leukemia. Simon's design is one of the most commonly used designs in Phase 2A clinical trials. Phase 2A is where we want to establish a proof of concept that our treatment is effective. And Simon's design is an exact procedure that allows us to make that evaluation. The main advantage of the Simon's design is that it is exact and that it also allows us to stop the trial early if there is very strong evidence that our proposed treatment is likely to be ineffective. In other words, stop for futility. Phase 2 is the most likely phase at which a proposed treatment is likely to fail to show sufficient evidence to continue on towards being released as a treatment. And thus the ability to stop a trial early is important to reduce the overall costs of clinical trials at this stage. There are two types of criteria that can be used for a Simon's design: the optimum and the min-max. The optimum reduces the expected sample size of our design, while the min-max ensures us the lowest maximum total sample size for our study. In this particular study, the researchers had assumed that they were going to use the min-max criterion. So we'll select that from the design criterion row here. You will notice there that we can use the optimum, min-max, or we can see the equivalent sample size for a single stage design using the exact proportions test. In this case, they wanted a significance level of 0.05, and in their opinion, the maximum ineffective proportion would be 0.05. In other words, if they got a response of less than 5%, they would consider that to be poor, whereas they considered the minimum efficacy proportion to be 0.3. In other words, if the proportion of responses was greater than 0.3 or 30%, they would consider that good. They targeted a power of 80%, and using the default search criteria, the sample size is equal to 14, with two stages of 7 and 7. You can see here that at the first stage, after 7 subjects have been completed, if there had been 0 successes, then we would stop the trial early. And that the probability of that occurring, if the proportion was actually 0.05, would be around 70%. If we continued on to the final analysis, if the number of events was less than or equal to 2, then we would find against the drug being effective, or that it has a efficacy greater than 0.3. And therefore, but if the number of successes was greater than 2, we would, in that case, find for the drug and continue on to the Phase 2B or Phase 3 stage of our clinical trial. If we were to do the same design, but using the optimum criteria, the overall sample size would increase from 14 to 18. But if our drug is ineffective, we would expect a higher chance of stopping early, and therefore the expected sample size, if our drug isn't effective, would actually be lower. For an equivalent single stage design, the sample size required would also be 14. But in this case, we would have no chance to stop early after 7 subjects were accrued. In nQuery, we also cover a variety of other different designs that are used for Stage 2 clinical trials, including MCP mod, Fleming group sequential designs, one proportion group sequential designs, and many other one group designs for endpoints such as means and survival. To learn more, visit statsolves.com. Thank you for watching.